All right. Hello, everyone. Good afternoon. Welcome to lecture 39 of BMS 300. So as you all know, this is just going to be on PollEverywhere questions, and I will give you a chance to practice that, and then I will show you which slides those answers can be found in as well. And I believe later today there will be a practice exam released, right? So you can practice that as well. What else? So on Monday we'll do a similar thing for the respiratory system, and on Wednesday we'll move to blood and the white blood cells, right? So we'll move on to the next unit, and I think maybe 15 or the last 20 minutes of Wednesday I'll open up the time for answering any questions you have, right? So yeah, does it sound good? And if you haven't already done this, and you want to improve your performance, I would highly recommend writing out the answers for the key questions. So get a notebook, use two different colors, one for the question, write down the answer, next question, write down the answer. It'll take time, but I've been noticing that it's paying off, right? From some of you who have been doing it, and you've been showing it to me, and I can already see an improvement in your comprehension of the material. So for those of you who haven't tried this out, please try it. It may change the way you study. Okay, with that, are there any questions for me before we get started? Nothing at all? Okay, all right, let's get into a lot of questions then. Okay, so the first question is, a PCA value of 4.0 indicates a lower calcium concentration than a PCA of 5.0. True or false? Is it locked? Oops. So eventually I'll be asking you to compare PCA 50 values between a wild type and a mutant, and then you could make a decision based on calcium sensitivity. But before we get there, you should be able to know what does a low PCA value versus a high PCA value mean, right? What has a higher calcium concentration than the other? Okay, we have a lot of responses. Good. So most of you said false. So a PCA value of 4.0 is referring to a higher calcium concentration compared to a PCA value of 5, right? And what does the word PCA stand for? What is it? Yeah, it is the calcium. Thank you. Negative logarithm of the calcium ion concentration. Just like how, what does pH stand for, right? Negative logarithm of the hydrogen ion concentration. So last class someone said PCA is referring to the partial pressure of calcium. That is not the right answer. I might use it as an option moving forward, right? I know you've seen a lot of small p and O and CO2, and so it's not the partial pressure, right? So PCA is referring to the calcium concentration. And if you look at this figure here, notice that the calcium concentration is starting from a low level and it's increasing. And as the calcium concentration is increasing from 6.5 to 4.5, the amount of tension produced is actually increasing, because when you have more calcium, what happens at the sarcomere level? What happens when you have more calcium present? Yeah, what is it? More calcium binds to what? Binds to something, and then what happens? Wait, it binds to TNC, right? And as a result, it leads to interaction between actin and myosin, right? And that leads to force production. So as the calcium concentration is increasing here, I hope it is clear to you why the force produced is increasing on the y-axis, right? And so when we talk about the next concept of PCA 50 and calcium sensitivity, you always have to compare between the wild type versus the mutant, right? The PCA 50 value by itself doesn't mean much unless you can compare it with the wild type and see if there's an increase or decrease in calcium sensitivity. So in that regard, let's look at this question. So if the PCA50 value for a wild type is 5.6 and for the mutant is 5.8, then which of the following statements is correct? The wild type has a higher myofilament calcium sensitivity than the mutant, or does the wild type have a lower myofilament calcium sensitivity than the mutant? Can you see how I'm ramping up the complexity? Unless you know what PCA stands for, you can't answer this question. So let's break this down. What does myo stand for? Muscle, filament, fibers, something very thin, right? So myofilament is those thin structures found inside the muscle, but it is collectively referring to two types of filaments we talked about. Thick and thin filament, right? So the thick and thin filaments, they are collectively known as the myofilaments, and the question is how sensitive are they to calcium, right? So if they have a higher calcium sensitivity, it should produce greater or less force for a given amount of calcium, greater amount of force, right? So let's see what the class said. So most of you said that the wild type has a lower myofilament calcium sensitivity, right? And that is the correct answer. And let me explain why. So we see that you measure PCA50 at the 50% of the maximal force generated by both fibers, right? So the amount of force is both at 50%, and for achieving that 50% force, we see the wild type is requiring a PCA of 5.6, which is higher calcium than a PCA of 5.8. So the mutant is only requiring less calcium to get to that 50% force, whereas the wild type is requiring more calcium, and that's why it has a lower myofilament calcium sensitivity than the mutant. Is that clear, folks? Okay, please make sure you understand this concept, because this is something I promised to test you on, right, on Wednesday. So if it isn't clear, please ask me now. Okay, let me just, yes? Good question. Why is the lower number indicate a higher concentration? For this very reason. It's the negative logarithm of the calcium ion concentration. And that's why a lower number is indicating a higher concentration. Is that clear? So let me just walk you through this slide again, just to refresh your memories. Right? I told you that the way they measure calcium sensitivity in the lab is like this. So they would take the muscle fiber and put it in calcium solutions of different concentration, right? That's what this cartoon here is indicating. And then as you increase the calcium concentration, so you put it initially in a PCA 9.0 solution, that means there's literally no calcium available, and you see there's no force produced. Then you increase it and the calcium concentration to PCA 6.0, and then it gradually goes up to PCA 4.3, and the amount of force produced when the fiber is in these different calcium solutions, you see it is gradually increasing, right? And then they take all of these values and they plot the force generated on the y-axis and the calcium, the corresponding calcium concentration at which they generated that force on the x-axis, and that's how they come out with this force PCA tension relationship. Okay, so that's how we see that the PCA50 is the calcium concentration required for the fiber to achieve 50% of its maximal force. And this concept will also tie into the leftward shift versus the rightward shift, right? And when you see a question in the exam which is telling you the PCA50 of a wild type is, let's say 5 and the mutant is 5.8, then you should be able to draw this figure out, and then it'll make sense, you know, whether there's increased or decreased calcium sensitivity. Is that clear? All right, so think about this. If you still need more clarification, we can talk about it on Monday as well, all right, on a one-to-one basis. So muscle relaxation will occur when which of the following takes place? Is it when the inorganic phosphate is released from the myosin head? Is it when ATP displaces ADP from the myosin head? When TNI binds to TNC? So we will have challenging questions like the one shown in the previous slides, but there will also be some easy questions like this as well, right? It will be a mix of everything. Okay, most of you said when ATP displaces ADP from the myosin head, and that is correct. And this is based on the cross-bridge cycle, right? And we see that the myosin head is detached only when ATP binds to the myosin head and causes the ADP to be displaced. So this is what causes the myosin head to detach. But then in order for the myosin head to attach weakly to the thin filament, there has to be calcium, which has to interact with TNC, and then that binds with TNI, and that whole complex moves. That's when there's weak interaction. But then for the strong interaction of myosin head with actin, you need to have the inorganic phosphate being released, right? So this is a slide where you will need to know what is leading from one to the other, right? What causes detachment, what causes weak binding of the myosin head, and what causes strong binding of the myosin head, and what makes the strongly bound myosin head to detach, and so on. You remember we even talked about this concept in regards to rigor mortis, right? You remember that? Okay, great. So we see the calcium influx to balance the calcium efflux is responsible for which of the following? The repolarization of the action potential, depolarization of the action potential, the refractory period of the action potential. Is it locked? Oh, okay. So what action potential is this referring to? What type of cell will you find this kind of action potential? What is it? It's a contractile cell in the heart, right? Not the autorhythmic or the skeletal muscle cell. Okay, most of you said the refractory period of the action potential, and this is correct. So this question was based on the action potential in the cardiac contractile muscle cells, and we see that for the initial depolarization to take place, you need sodium to come into the cell, and then there is this plateau phase, because as potassium is leaving the cell, calcium is going to come into the cell, and that's why the membrane potential is being steady. You have this plateau phase, but then after some time these calcium channels will close, and so there's no more calcium influx, and so there will be rapid repolarization as the potassium is leaving the cell. Is this clear? The goal I have for you is to be able to explain why each of these phases of the action potential are taking place, right? And also an interesting thing I mentioned in the slide is that there is calcium-induced calcium release occurring in the cardiac muscle cells. So the calcium which is coming into the cardiac myocyte and causing this plateau phase, that calcium can go in turn cause the release of calcium from the sarcoplasmic reticulum, and this additionally released calcium can further ensure force production, right? So this is a unique feature which is found only in the cardiac muscle cells. It's not there in smooth or in skeletal muscles. Okay, moving on. Here's another simple question on the chambers of the heart. So which of the following chambers is going to receive oxygenated blood? Is it left ventricle, right ventricle, left atrium, right atrium? Okay, most of you said left atrium, and the answer for this slide is right here. So left atrium receives oxygenated blood from the pulmonary veins. Is this clear, folks? So there will be some simple questions too, but I hope you are ready for most of them, right? That's why I tell you to do the key questions. Okay, the rapid depolarization of the pacemaker cells is due to blank. Is it due to calcium channels, sodium channels, HCN channels? So this is talking about the pacemaker cells and their action potential and what is causing those various phases of that action potential. Man, everyone thinks it's HCN channels. HCN channels do play a role in the pacemaker cells, but the right answer is calcium channels, right? So the question is, what is causing the rapid depolarization? And that is due to calcium influx. And what causes the calcium influx is the opening of the voltage gated calcium channels when this threshold is reached. And what causes the threshold to be reached is the HCN channels. And HCN stands for hyperpolarization cyclic nucleotide. Fantastic. So you need both this hyperpolarization of the membrane potential and cyclic AMP binding to the HCN channels, and that causes them to open and sodium can come into the cell, and as sodium comes in, this threshold is reached, causing the voltage gated calcium channels to open. Are we clear, folks? All right, moving on. Decreased blood being brought to the myocardium is a direct consequence of which of the following? Is it a cardiac arrest, heart attack, arrhythmia? So when which of these happen, will there be reduced decrease in blood brought to the myocardium? So myocardium is referring to the walls of the heart, and the question is, when will blood not be brought to the myocardium? Okay, most of you said heart attack, and that is correct, right? So heart attack is this problem where the blood vessels are blocked, and so you're not having sufficient blood being delivered, right? Whereas cardiac arrest is where there's an electrical problem, and that can lead to irregular heartbeat and arrhythmia and so on. And any questions at all, folks, on this? All right, great. Moving on. Abnormal T waves in the electrocardiogram can be most likely indicative of defects in what? So if you have an abnormal T wave, is it a problem in the ventricular systole, atrial systole, ventricular diastole, or is it something else? Okay, most of you said ventricular diastole, right? So this is the slide from which you can find the answer. So you see that the T wave is going to indicate the repolarization of the ventricles, and the ventricles repolarize, it will lead to ventricular relaxation, and the contraction is called systole and relaxation is called diastole. So a problem in the T wave is going to indicate problems in the ventricular diastole. Similarly, I hope you know what the P wave and the QRS complex stands for as well, right? Yes?
What part of the conduction system is T wave?
Oh, what part of the conduction system? It's going to happen throughout, right? So the T wave is just referring to the repolarization of the ventricle, and the way they come up with this electrocardiogram is they have the electrodes positioned in a very specific way so that when the atria is depolarizing, it leads to the P wave, and if the electrodes were not positioned properly, they won't generate this type of wave pattern. So the electrocardium is just the collective electrical activity of the myocardium. It's just not the conduction system alone, it's the electrical activity throughout the heart. And keep in mind that all of the cells are connected by what junctions in the heart? gap junctions, right? That's how the electrical currents can pass from one cell to the other. Okay, good question. So abnormal QRS complexes in the electrocardiogram can be most likely indicative of defects in what? Is it ventricular systole, atrial systole, ventricular diastole? So you see how I modified the previous question? I just asked you about a different part of the electrocardiogram. Okay, most of you said ventricular systole. Good. I'm glad you're catching on. So here's another figure I used to explain it. So we find that the P wave is going to indicate the atrial depolarization, and as a result the atria will contract, and then the QRS complex is indicating the ventricular depolarization and the ventricle will contract. But while the ventricle is contracting, the atria has already repolarized and is relaxing, but there is no specific wave in the electrocardiogram to indicate the atrial repolarization. And then when we come down to the QRS complex, sorry, when you come down to the T wave, not to the QRS complex, when you come down to the T wave, the ventricle is repolarizing and it is going to relax as a result. And I hope you know what each of the waves and the complexes in the electrocardiogram stand for as far as the cardiac cycle is concerned. Alright, so the next question is, the slope constructed during the end systolic volume in the pressure volume loop indicates which of the following? Does it indicate the afterload contractility or the preload? So you have the pressure volume loop and then there's a slope constructed at the end diastolic volume. What is that indicating? Ooh, the answer is all over the place. So I wanted to say the minorities, right, but someone said I don't know. So the right answer is actually contractility, right? So option B. But you could answer this question if you were able to explain what's in this graph, right, in this figure. So let me walk you through the pressure volume loop. We find that pressure is shown on the y-axis, and on the x-axis we have changes in volume, and this point is indicating the end of systole, right? And this question was asking you about the end systolic volume and the slope drawn there, right? So the slope drawn at the end systolic volume is right here, but then we see that the end systolic volume is shown at this place as well. So at this point we find that the atrioventricular valve will open because the pressure in the ventricle is going to drop below the atrial pressure, and so the AV valve opens, and when the valve opens, blood from the atria can flow into the ventricle, and as the blood is entering the ventricle, notice that the volume of blood in the ventricle keeps increasing and it reaches this end diastolic volume. So end diastolic volume is when the ventricle is fully relaxed and it is filled with blood. And this point where the where you have the end diastolic volume, it's also known as the preload, because preload is indicating the length at which the muscle has been stretched to before it can contract. And at this point, notice that the ventricular pressure starts increasing, and the reason it increases is because the QRS complex has taken place, the ventricle has depolarized, and then it starts to contract. But at this point you have isovolumic contraction, because the ventricular pressure, while it is higher than the atrial pressure, is still lower than the aortic pressure, and that's why there's no change in the ventricular volume, no blood can be pumped out, and so you have this isovolumic contraction, and at this point the ventricular pressure is overcoming the aortic pressure, and then the aortic valve opens. So this here is the afterload, the pressure which the ventricle has to overcome in order to eject the blood. So this is the afterload, and we see blood is being ejected out, and as blood is ejected out, we see that the volume decreases, and we find that the pressure also gradually decreases because the T wave has taken place and the ventricle has repolarized, right? So the ventricular volume decreases, and at this point it drops below the aortic pressure. So the aortic valve closes, and then you have the isovolumic relaxation. Now the slope drawn at this end systolic volume is going to indicate inotropy, or in other words, contractility. So it's just indicating what is the amount of pressure which can be generated by the ventricle at a given muscle length, right? And the steeper this slope is, it means there's a higher contractility. The more shallow the slope is, it means there is less contractility, right? And I even showed you a picture in the slides where how the contractility would change, and when contractility changes, it will in turn have an impact on stroke volume and how much blood is being pumped out of the heart. Is this clear, folks? The pressure volume loop. I really hope that you're able to draw out the pressure volume loop, because when there's a question like this on the exam, unless you can draw it out, then it'll be very hard to answer it, right? That's why I recommend you to do the key questions, so write it out, right? Okay, moving on. Here's a question about the cardiac output, right? The cardiac output is inversely associated with end diastolic volume, end systolic volume, heart rate, and stroke volume. So what is it inversely associated with? So in other words, you can also interpret this question as cardiac output would decrease if there is an increase in end diastolic volume, or if there's an increase in end diastolic volume, or increase in end systolic volume, or increase in heart rate, or increase in stroke volume. Right? That's another way you can think about this question. So increasing which of the following will cause a decrease in the cardiac output? Okay, most of you said end systolic volume, and that is correct, but let me explain it for the rest of you, right? So we see that the cardiac output is determined by two factors, right? What is it? Looking at the slide, is dependent on, you can look at the slides and tell me the answer. Dependent on heart rate and stroke volume, right? So we see the heart rate is going to be influenced by the autonomic nervous system and dependent on what hormones are released, like epinephrine, norepinephrine. Whereas stroke volume is indicating, okay, before we go to stroke volume, heart rate is just the number of beats per minute, right? The number of times the heart will contract and relax per minute. That's the heart rate. Now stroke volume is referring to the amount of blood ejected when the ventricles contract. I repeat, the stroke volume is referring to the amount of blood ejected when the ventricle contracts. So to calculate the stroke volume, you can subtract the end diastolic volume, the volume of blood in the ventricle when it's fully relaxed, minus the end systolic volume, the amount of blood remaining in the ventricle after it is contracted, right? So if you subtract the two, you get stroke volume. Now if you have an increase in stroke volume, it will increase the cardiac output, because the larger the stroke volume, it's going to increase cardiac output. If you increase the end diastolic volume, that will also increase the stroke volume and thereby affect the cardiac output. But if you have a higher end systolic volume, which means like after the ventricle has contracted, if you have more blood remaining still in the ventricle, that means you have less cardiac output. It means less blood is being ejected out, and so the cardiac output will go down. Is that clear, folks? Right, so this is an important relationship. I hope you know which one has a direct proportionality to the cardiac output versus the inverse proportionality. Okay, the autonomic nervous system can blank heart rate by causing the blank of the membrane potential of pacemaker cells. Right, so the blanks could be increased repolarization, decreased depolarization, increased repolarization, and so on. So whenever you see a question like this, right, if you look at the question and then try to focus on the answers, you can get easily thrown off, right? So think about what this question is referring to, what graph or figure you'll have to draw out, and once you draw that out, you fill up these blanks and then you pick the right option. It's kind of like fill in the blanks, but just looking directly at the options may throw you off. Okay, most of you said option C, and that is correct. Right? Let me walk you through this, how you can have a increase in heart rate by causing decreased repolarization. So this is the slide which should have come to your mind, right, when you read that question. So notice that when you have the parasympathetic stimulation, you have a greater degree of repolarization or hyperpolarization. So the greater the hyperpolarization is going to cause the threshold to be reached at a slower rate, right, because it is more farther from the threshold. It's going to slow the heart rate. Whereas if we have the reduced repolarization after the initial depolarization, then the threshold can be reached a lot quicker, right? And we see that the sympathetic nervous system, when you have epinephrine and norepinephrine released, they will cause this reduced repolarization by opening both the calcium and the sodium channels. And so that's why you have this reduced repolarization. Right? Is that clear, folks? All right. So moving on, which of the following permit the maximum exchange of substances by slowing blood flow to the greatest extent? Is it when blood flows through the elastic arteries, veins, arterioles, or muscular arteries? So to answer this question, you need to know the order in which the various blood vessels branch, right? You see that the aorta takes blood out of the heart, the vena cava brings blood to the heart, but how is the aorta branching eventually to form capillaries, and how is the capillaries leading eventually to the vena cava? If you can understand that, then you know how the pressure will change in the blood vessels, and also the flow will change as well. Okay, most of you said arterioles, and arterioles is correct, right? So, okay, let's look at this slide here. So you can answer this question if you were able to remember what was on this slide. So let's start with this figure C, right? So notice that on the x-axis you have elastic arteries which give rise to muscular arteries, which give rise to arterioles, which give rise to capillaries, venules, veins, and vena cava. The order of these blood vessels is really important that you know that correct order, right? And notice that as you go from elastic arteries down to vena cava, the blood pressure is dropping dramatically, right? And we see that the steepest place of blood pressure drop is in the arterioles. And we find that as the blood pressure is dropping, you find that the velocity of flow is also decreasing in the arterioles, right? As the pressure decreases, the velocity of flow is going to decrease, and it reaches a minimum at the arterioles and capillaries, so that the slower it is moving across in these vessels, there can be greater exchange of substances like gases, nutrients, wastes. All of that exchange can take place, right? And there is also another important concept to keep in mind, that even though this velocity is decreasing, gradually there seems to be an increase in velocity as the blood moves from the capillaries to the vena cava, right? And the reason for this increase in velocity is that in all of these blood vessels, right, the arterioles, capillaries, they are going to be more in number than the veins and the vena cava, and so you're going to have a larger overall surface area, cross-sectional area, and when you have blood moving from a greater cross-sectional area to a smaller cross-sectional area, the velocity of flow is going to increase. Just like how if you have water poured into a funnel, as the water travels down the funnel, it's going to travel a lot faster because you have blood flowing in from a larger cross-sectional area to a smaller cross-sectional area. So is this clear, folks? And I hope you know what you need to memorize, the order of the blood vessels. Okay, so which of the following pathological outcomes can occur rapidly and without warning? Is it a heart attack? Is it a cardiac arrest, atherosclerosis, or coronary artery disease? So if you want a hint to remember this, think about the slide I showed you on playing Monopoly and going to jail versus playing a game where you have a siege on a castle and it's being attacked. Okay, most of you said cardiac arrest. So, and that is correct. So cardiac arrest is what can happen rapidly without warning. So cardiac arrest is an electrical problem, whereas a heart attack is the blood vessels being clogged and you have less blood being delivered. And atherosclerosis is just referring to the buildup of plaque on blood vessels. It can happen all over the body, right? Whereas the coronary artery disease is where you have the blood vessels in the coronary artery. So these are the blood vessels of the heart which are clogged, and so blood supply to the heart is going to be affected. So is this clear, folks? Why cardiac arrest is the right answer? And here's the slide I walked you through in class. Okay, moving on. Ventricular filling is complete immediately after which of the following? Is it after the T wave, the QRS complex, P wave, or the P to T interval? So this is again tying back the electrocardiogram to the different parts of the cardiac cycle. We have over 60 responses. Okay, I know it when the class is struggling with it, because when the answers number doesn't increase very quickly, you see people are thinking about it. Well, the correct answer is P wave. So we find that the most of the ventricular filling is going to take place during the T wave, right? When the ventricle is has repolarized and it's relaxing, most of the ventricular filling takes place. But the final bit of filling takes place when the atria contracts and pushes in the last amount of blood, right? The last bit of blood, and that happens after the P wave. So the ventricular filling is completed immediately following the P wave. Is that clear, folks? Are these questions too easy? Are they too difficult? Are they somewhere in between? It seems like achievable, right, if you focus on these concepts. All right, great. I want you to walk out of this room with hope, right? So again, so here's the slide which I hope you are able to explain, right? So we see that the P wave is causing the atrial depolarization, where you have the contraction and that forces blood into the ventricles, and that's when the ventricular filling is actually completed. And following that, you have the QRS complex, where the ventricles will depolarize and then they will contract and force out blood, right? And finally, after the QRS complex, we have the T wave, where the ventricles will repolarize and relax. Okay, moving on. In the pressure volume loop, a decrease in the end diastolic volume is most directly indicative of a decrease in blank. Is it contractility, inotropy, preload, or afterload? So if the end diastolic volume decreases, it's most likely indicative of a decrease in which of the following? Okay, most of you said preload, and that is correct. Again, we come back to this slide, right? I really hope that you're able to draw this out from memory, right, where we talk through the various parts of the pressure volume loop, and end diastolic volume is here, and this represents the preload, the preload, not preload, the preload, right? Okay, great. This is clear, folks? All right. Okay, moving on. In the skeletal and cardiac muscles, calcium is stored where? Is it in the sarcoplasm, smooth endoplasmic reticulum, sarcoplasmic reticulum, or Surca pump? You can see people are pretty confident. The number is increasing pretty rapidly here. Okay, most of you said sarcoplasmic reticulum, and that is correct, right? So, for those of you who had sarcoplasm is just referring to the cytoplasm inside the muscle cell, right? And the smooth endoplasmic reticulum is not the right term. Sarcoplasmic reticulum is a type of smooth endoplasmic reticulum which has been modified for the muscle cell, right? And what is the Surca pump, right? So let's look at this picture here. So sarcoplasmic reticulum is shown in this blue box. Surca pump is what takes calcium back into the sarcoplasmic reticulum. That's what the Surca pump does, right? And you have the dihydropyridine receptors which are activated as the action potential goes through the T tubules, and the ryanodine receptors are the ones through which the calcium is actually released out of the sarcoplasmic reticulum. Is this clear, folks, how this question was based on this slide? Okay. All right, moving on. The cardiac muscle can exhibit the Frank stalling mechanism where the muscle length determines the force generated. The reason for this is that physiologically the cardiac muscle is present in the blank of the force length relationship. So is the cardiac muscle functioning in the descending limb, ascending limb, plateau region, or the region of maximal overlap between the thick and thin filaments? Where is the cardiac muscle functioning? Okay, we have, so most of you said ascending limb, and that is correct, right? For the rest of us, notice that this is the figure from which I asked this question, right? So we find that the cardiac muscle is functioning in the ascending limb. So even for a small change in muscle length, you will have a dramatic increase in force, right? The percentage of force increase is greater in the cardiac muscle, so it is more sensitive to changes in muscle length. Whereas the skeletal muscle, it's going to produce more force because it's found in this plateau region, right? So it will generate more force, but changes in muscle length will not result in as dramatic of an increase in force. So the percentage change in the skeletal muscle won't be as large as in the cardiac muscle. Okay. Moving on, the threshold for rapid depolarization in the autorhythmic cells is reached by the activity of what? Yes, what's your question? Oh, it is just the name given, right? This is called the ascending limb. You could call it the ascending face if you want. It's called the ascending limb. And then you have this descending limb and the plateau region. If you want to call it ascending region, descending region, that's fine too. But in science we have certain terminology and we choose to stick to that, right? Okay, moving on to this question, the threshold for rapid depolarization in autorhythmic cells is reached by the activity of what? I already showed you the slide based on which this question was asked before. Okay, most of you said the HCN channels, hyperpolarization, cyclic nucleotide gated sodium channels. So that is the correct answer. So we find that in order to reach the threshold, you need to have sodium influx, and that is due to the H-scene channels being activated. And what activates the H-scene channels is this initial hyperpolarization and also the binding of cyclic AMP as well. Okay, moving on. During isovolumic relaxation, there is no change in volume because the ventricular pressure is blank than the aortic pressure but blank than the atrial pressure. So is it higher, higher, lower, 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 higher, higher, lower? What is it? So again, in questions where you have multiple blanks, try to fill it out by yourself and then look at the options. Okay, the right answer is C. The ventricular pressure is lower than the aortic pressure, but it is higher than the atrial pressure, right? And so I really hope that you're able to draw this figure, right, from memory. And if you haven't been able to do it till now, I would highly recommend that you can. So the top panel is showing you the electrocardiogram and the corresponding changes in the pressure in the atria and in the ventricles in the second panel, and then the change in the volume of blood in the ventricles, right? And so this question was asking that during isovolumic contraction or relaxation, why is no blood flowing out, right? The blood is not flowing out because this atrioventricular valve is closed and this aortic valve is also closed. Both are closed, and in order for this AV valve to be closed, the ventricular pressure must be higher than the atrial pressure, and for this aortic valve to be closed, the ventricular pressure must be lower than the aortic pressure, right? So is that clear, folks? Okay. All right, with that we finished 2 minutes early. If you didn't get the answers right on this, please go ahead and work on at least these slides which I showed in class today and be able to write it from memory. And I will see you on Monday, where we will do the PollEverywhere questions for the respiratory system. Have a great weekend, everyone.